Have you ever wondered why humans just seem to crave sugar? It's almost as if we need it to survive. In this video, we'll take a look at glucose, sugar, and why we need it. Let's get started. So there's an amount of free energy needed to survive. Living organisms need energy. And we get that energy in different ways. If you're an autotroph, like the grass here, they capture their energy from the environment. So we have the sun, photosynthetic energy coming down, photosynthesis occurs, energy is available for the plants. There are some small organisms that actually use little chemicals. We call them chemosynthetic organisms. They make their energy from those chemicals. So autotrophs, think auto, self, they make their own energy from the environment. Now, what about the rest of us? The rest of us are heterotrophs. We need other things for our energy. So we get energy in carbon compounds, right? Carbs, lipids, proteins. We break those down, we hydrolyze them to get free energy. And of course, you can see the grasshopper is an example of a heterotroph. So the focus of today's video is really about heterotrophs. How do heterotrophs get, utilize, and manufacture all the energy they need? So when we talk energy, we often talk about a molecule called ATP. ATP is our energy currency, right? You think of currency, right? You've got some cash, you want to go buy something, you use your currency, you turn it over, you get something in exchange. Well, ATP is sort of our energy currency. It's what we use to get work done in the cell. So ATP stands for adenosine triphosphate, ATP. This is really just a modified nucleotide. So we have an adenine, which is a base, a ribose sugar, and then several phosphates. Now, AMP stands for adenosine monophosphate. So that would be if there was the adenine, the ribose, and one phosphate. When you add another phosphate, so you take AMP plus a phosphate, you get ADP. That would be ADP. When you add that third phosphate, right, so you take adenosine diphosphate and add the third phosphate, you will get ATP. So here's our molecule. We see the adenine here, we see the ribose sugar there, and the three phosphates. So maybe you're asking, okay, ATP, it's our energy currency, but how does a molecule store energy? Well, take a look here. Here's AMP, right? AMP. Adenosine, there's our adenine, our ribose, and our 1-phosphate. How does it store energy? Well, every time we add a phosphate group to this molecule, as you can see there, look, you'll notice these negatives, right? And we know that negatives and negative and negatives will repel each other, right? So as we put more negatives and we pack them, sort of like spring loading them, what happens is there is energy stored in this molecule now, because these things really are like a spring. The more you push together, right, those negatives, they don't want to be close, so they're ready to fly off and do some work. So when we add that third phosphate, now we have ATP. These electronegative phosphate groups are difficult to add. So once they get added, then they're essentially available to do a lot of work when they leave, when the phosphate group leaves. So these negatives packed closely together, sort of think of it like a spring. Once one of the phosphate groups flies off, you'll see that the energy is available. And that's exactly what I want to show you here. So how does the ATP transfer the energy? We call this phosphorylation, right? Because a phosphate group is going to leave and go to another molecule. So ATP will take its third phosphate group and it will add it to another molecule. And when it does that, it will release energy, transfer it to the other molecule, and it will be able to have some work, some sort of change that it needs. So when that third phosphate group leaves, notice that it now takes with it or lets off some energy available for that other molecule. So when ATP is fully loaded, it essentially has stored energy available to transfer. So this cycle, I think, will make sense to you now. So if we have ATP and we lose a phosphate, right? So there's ATP, there's its 1, 2, 3 phosphates. If we lose a phosphate, it will go down to ADP, right? So you've got the 2 phosphates there plus that phosphate group that left. Now, then what happens? Well, then we need to go back and we need to regenerate, make more ATP, so that that ATP can be used to do some things in the cell. Then that ATP, of course, will go back, need to be regenerated ATP, and the cycle continues. This is an important cycle because if you think about a working muscle, right, a muscle that you're using actively, actually requires and recycles about 10 million ATPs per second, right? That's a lot of ATP, a lot of molecules. So this recycling of this energy currency is very important. So where do we get the energy? I said we need to make all this ATP. How do we make it? Well, actually glucose, sugar, right? Remember I started talking about sugar. Sugar is how we make ATP. So we catabolize or break down glucose and we make ATP. This process is called cellular respiration. This is the process of taking glucose and oxygen and through several steps converting it to carbon dioxide, water, ATP, and then of course, as always, a little heat is given off. This process called cellular respiration is very detailed. Many people have a hard time understanding it. I'm going to do my best to start with you today. We'll go over the overview and then we'll go through the steps of glycolysis. But basically you just take glucose and oxygen through a series of steps. We get carbon dioxide, water, ATP, and some heat energy. Before we can do this, we have to talk how electrons are moved. Part of this process that we're going to see is about moving electrons. We call these reactions oxidation reduction reactions. And electrons, right, if you think of, electricity, electron flow, well, in physical things in space and things that we've manufactured, right, electrons can move down a copper wire or something. But in the body, the electrons have to move as part of a hydrogen atom. So when they move, we have these oxidation reduction reactions occurring. And I have a little thing that helps me remember oxidation and reduction. If something loses electrons, that is oxidation. If something gains electrons, that is reduction. So the little device is Leo says grr. Leo the lion says grr, right? That might help you remember how this works. So if you take this molecule, whatever it is, if it loses electrons, right? Losing electrons is oxidized. Those electrons have to go with a hydrogen. Remember, electrons move as part of a hydrogen, and comes over to this molecule, which is gaining electrons. So if we think about it, this molecule here, because it lost electrons, has been oxidized, and that's what you see there. This molecule over here, because it gained electrons and the hydrogen with it, gaining electrons has been reduced. So we see the reduction taking place there. Now what's that mean for the charge? If something lost negative or lost electrons, it now has a slightly positive charge. If something has been reduced or gains electrons, more negative, then it will have a negative charge. So this is an oxidation and a reduction reaction. So I told you these electrons move with hydrogen atoms. They move also by electron carriers. So electron carriers move the electrons by shuttling hydrogen atoms around. And these are the two electron carriers that we're going to talk about, NAD and FAD. When those are in this form, that's their oxidized form. But when they're reduced, right, gain electrons, when they gain the electron and the hydrogen, this is their reduced form. Notice that they have these H's as part of it. So NADH, FADH2 are our electron carriers. And here's just sort of what you see. Here's NAD, which happens to be vitamin B3. That is one of our electron carriers. Notice it has a slightly positive charge here on this particular end of the molecule. So when this becomes reduced, a hydrogen with its electron, remember, is added. Reduction occurs, and that hydrogen attaches to the molecule, and now it no longer has that positive charge because it carried an electron with it to help the charges balance. Now the opposite can take place, of course, and the oxidation can occur, and we can go back to this step, right? So these electron carriers just go back and forth, right? Takes NAD, picks up the hydrogen electron, takes it over. Once the electron is used, it'll go back. So much like ATP, electron carriers go through their own cycle. So let's do an overview of cellular respiration here. There's the equation again, right? And there are 4 stages of cellular respiration which I want to talk about. The first stage is anaerobic respiration or glycolysis. So when you think aerobic, you think oxygen, like you're doing some aerobics, processing a lot of oxygen. Anaerobic, anytime you put an in front of it, that means no oxygen. So the first stage of cellular respiration does not require oxygen, and that stage is glycolysis. Glycolysis occurs in the cytosol, right? So here's our cell. Here's inside. Here's the cytosol, or inside. And notice that glycolysis just takes glucose, breaks it down to pyruvate. In that process, we're going to make a little ATP. We're going to make a little energy. We also, in that process, are going to reduce NAD and turn it into NADH. That will then carry those electrons over to a final step that we're going to see in a moment. Okay? So step 1, no oxygen required, glycolysis in the cytosol. Then we have a choice. There's a fork in the road. If oxygen's present, pyruvate, this 3-carbon sugar, will go into the mitochondria, powerhouse of the cell, but oxygen has to be required. Oxygen has to be present. This is aerobic respiration, and the final 3 stages of cellular respiration use aerobic respiration. So if oxygen's present, the pyruvate will be oxidized, come into the mitochondria. If there's no oxygen, that pyruvate will go through a process of fermentation, which we'll mention in the next video. So if the pyruvate does go in, oxygen's present, we go in, pyruvate oxidation occurs. Then it will head on over to the Krebs cycle. The Krebs cycle, of course, will make some ATP, also regenerate some NADH, get some electron carriers for us, and those will ultimately all be shuttled over to the final step, the electron transport chain and oxidative phosphorylation. This final step is where we're really going to get the bang for our money. So this final step is where we're going to get a ton, this one over here is where we're going to get a ton of ATP produced. Okay? So there's your overview. There's your 4 stages. Glycolysis, no oxygen required, but these 3 over there do require oxygen for them to work. So we're going to first focus, in the rest of this video, we're going to focus on the first step, glycolysis. So when you think glycolysis, you think breaking down glucose. Glyco, right, sugar, lysis, breakdown. So glycolysis is just taking a molecule of glucose, that sugar that we all love, and then breaking it down through several small steps so that we can make ATP from ADP. The end result will be a pyruvate molecule from the glucose. So this is just a sort of general overview. Glucose, as we know, is a 6-carbon sugar, right? C6H12O6. So there's our 6 carbons. Through a series of steps, we'll break it down to fructose 6-phosphate, or we'll actually do some rearranging. That requires ATP energy. Wait, Herder, I thought we were making ATP energy. Yes, we are. But we actually have to put in a little activation energy first. We got to invest a little ATP so that we can get a bigger payoff of ATP in the end. So we've invested a little ATP. That ATP went down to ADP. And you see that we're now left with fructose 6-phosphate. That fructose 6-phosphate, through several steps, will eventually give us PGAL, or something we call G3P. That's an important 3-carbon sugar, 1, 2, 3-carbon sugar that's going to be responsible for doing several important things later. And then we'll see when we do photosynthesis, this is when we're building back sugar, the intermediary step as well. Then here's the energy payoff phase, right? So going from G3P through many, many steps, we're going to finally break it down to this 3-carbon pyruvate. But notice what we pick up. We're going to pick up some electron carriers, 2 and ADH. We're also going to pick up 4 ATP. Those 4 ATP will be used for the cell. The NADH is going to be shuttled over to be used in the final step, electron transport chain and oxidative phosphorylation. So what I'm going to do now is I'm going to go through glycolysis step by step. This is dense, this is heavy work, it's heavy brain, mental lifting. Try to follow along as well as you can. This is a very detailed process, and I'm just going to move through it so you can see what's actually happening at each step. So the first half of glycolysis is what I'm going to do on this slide. So we start with glucose, right? 6 carbons, 1, 2, 3, 4, 5, 6. You can see them all labeled there. Each of these steps uses an enzyme. Notice here, the first step uses hexokinase. You can see ASE, that helps tell us it's an enzyme. Remember, we also have to invest a little ATP. So what happens is, one of these phosphate groups from ATP goes down to ADP, and that phosphate attaches right here to the 6th carbon. So notice now that we have a phosphate group attached. So this is glucose 6-phosphate because the phosphate is attached to the 1, 2, 3, 4, 5, 6th carbon. Once we get glucose 6-phosphate, you'll notice that we have another enzyme, phosphoglucosaisomerase, which takes this molecule, glucose 6-phosphate, and then actually just rearranges it a little bit, right? We're not really adding a lot of stuff, but notice we went from this shape here to a smaller shape, and one of these carbons has moved out of the ring up here. So that is fructose 6-phosphate. So we've gone from glucose sugar to a fructose sugar. Then another enzyme, phosphofructokinase, actually adds another phosphate group. So here is again, we're investing, we're in the investment phase. ATP loses a phosphate. Now notice that it's attached there. So the phosphate group from ATP goes and attaches itself to that end. So now notice what I've added. Fructose 1,6-phosphate has a phosphate there on the 1st and the 6th sugar, so we call this fructose 1,6-phosphate. These names get pretty complex, right? Then we have this 6-carbon sugar is finally ready to split in half. So we take our 6-carbon sugar, basically it gets split in half by aldolase, and we end up with 2 different sugars. We end up with G3P, glyceraldehyde 3-phosphate, which is right there. Notice the 1, 2, 3 carbons. We also end up with dihydroxyacetone phosphate. Notice 1, 2, 3 carbons. Now, this one over here, G3P, is the only one that can move through the rest of the steps. So this one here, dihydroxyacetone phosphate, actually has to be converted by an enzyme called triose phosphate isomerase into glyceraldehyde 3-phosphate. Now that we have this molecule, G3P, we can go to the second half of glycolysis. So let me take you up there. So remember what we're leaving with. We're leaving with G3P. G3P now is acted on by glyceraldehyde 3-phosphate dehydrogenase. And if you think about dehydrogenase, right, what's going to happen? We're actually going to get some hydrogens now. So we took the molecule we had before, and notice that NAD came in, we added a hydrogen and an electron, okay? So here we've just made our electron carrier. So this molecule here is called 1,3-biphosphoglycerate. Then that molecule is where we're finally going to get some payoff. Because notice that we've got a phosphate there and a phosphate there. So we got 2 phosphates. But also remember Because we split the 6-carbon sugar in half, we now have 2 of all of these molecules. So I have 2 times biphosphoglycerate. So what's going to happen is phosphoglycerate kinase comes in, takes the phosphate group off up here. See this phosphate? Leaves, goes to ADP to make ATP. All right, there's our first one. But remember, because we got 2 of these molecules, we're actually getting 2 ATP made. All that converts to 3-phosphoglycerate. There it is. Notice we still got our phosphate group. So we're going to do a little rearrangement of this molecule by this enzyme, phosphoglyceromutase, to make 2-phosphoglycerate. Then this molecule loses some water through the enzyme enolase, and we're finally ready for our last step. This has been converted to this phospholinopyruvate. Notice it's still got that phosphate group we're going to get. Then we finally have pyruvate kinase, which takes the phosphate group, adds it to ADP to make ATP. And we're left with the famous pyruvate. And remember, all of this was occurring times 2. So we picked up 2 ATPs there. We picked up 2 ATPs there. So we essentially made 4 ATPs. Remember, though, we invested 2. So we really only net gained 2 through the process. Okay? So that's glycolysis. A lot going on. Hopefully you followed the flow. You saw as we went through it. I don't know that anybody truly has this memorized, so I wouldn't worry about trying to memorize this. But try to just understand the flow of it. So that last step, substrate level phosphorylation, is where a phosphate group transferred using an enzyme. This is called substrate level phosphorylation because phosphorylation happened, right? We had a phosphate group transferred, and we used an enzyme to do it, so that was at the substrate level. So this is a kinase enzyme. Remember, this was that last step. And what we saw was that this enzyme, pyruvate kinase, took the phosphate and added it to ADP. So it used an enzyme, pyruvate kinase. So essentially these fit as substrates, so we call this substrate level phosphorylation. Okay, let's wrap this up. Let's do some energy accounting. So remember, we started with glucose. We ended up with pyruvate at the end of glycolysis. We went from a 6-carbon sugar to 2 3-carbon sugars. We invested 2 ATP. We gained 4 ATP. So that means we had a net gain of 2 ATP. We put 2 in, we got 4 out. Net gain is 2 ATP. So this seems like a lot. That's a lot of work for the cells to do for only 2 net gain ATP. There's got to be a better way. And in fact, there is a better way. Remember those electron carriers? They're going to shuttle over to a final step, which we will look at in the next video. So take it all in. Glycolysis is a tricky thing to talk about, but remember the point. The point of all of this is just to make us some energy, ATP.